Okay, it is now officially 11:30, so sorry for the early start a moment ago. Welcome back, everyone. I hope you enjoyed your break. Um, it is my pleasure to welcome Carrie Tribble. Carrie is an evolutionary biologist who uses statistical methods to model the evolution of complex and understudied natural phenomena, particularly in plants. She received her PhD in integrative biology from the University of California, Berkeley in 2020. She is currently a postdoctoral researcher at the University of Hawaii in Manoa, and I'll let you correct that pronunciation if I pronounced it incorrectly, School of Life Sciences. And I'm sure if we were doing this conference in the middle of winter in Minnesota, we would all be even more jealous of you than we currently are. So I am going to mute myself and pass it on to Carrie.
Great, thank you so much for the introduction. And before I get started, I want to thank everyone who's been involved in planning and coordinating this conference and for inviting me to join. I am a postdoc researcher at a university, and so I spent a lot of time talking with other academics and other researchers, but it's been really lovely to be participating in this conference and hearing about how people in other aspects of society and in different industries are using R in their work, and thinking about the ways in which R can sort of span across all of these different goals as a really useful tool. So I've been really enjoying the conference so far. And with that said, I am a evolutionary biologist at heart. And so what I'm hoping to do today is to share a bit about how I use R and how my colleagues use R, and specifically touch on a recent R package that my colleagues and I have been developing. And so hopefully you'll come away from this presentation with just an appreciation for the ways that R is being used in academic research and evolutionary biology in particular. So I started out as a young student of biology, really inspired by this stunning array of natural variation that we observe across all living things. As an evolutionary biologist, I seek to understand the processes that generate this observed biodiversity. For example, we might wonder how closely this Laysan albatross is related to other animals, such as this coral. And it turns out that these two animals last shared a common ancestor over 800 million years ago. In other words, these two organisms have been evolving independently from each other for over 800 million years. And we may also wonder how these Hawaiian honeycreepers were able to evolve such different morphologies in their beaks. You'll notice some of these birds have really long curved beaks, and others have short, thin beaks. And it's a little hard to see here, but there's also a bird in the middle of the screen that has sort of a squat, fat-looking beak. And these birds have evolved all of these differences in their coloration and their beak morphology pretty recently. They all evolved from a single bird, common ancestor, that arrived to Hawaii around 5 million years ago. And these different beak morphologies allow these birds to specialize on different food sources, such as nectar from flowers, insects, and seeds. So the birds with the long curved flower beaks often feed on nectar from flowers that require these specific bill shapes in order to reach the nectar inside of the flower. So these are some of the questions that evolutionary biologists seek to answer. How did this stunning assemblage of biodiversity emerge? How are these organisms or other organisms related to each other? Why are some organisms so different while others are very similar? And how do organisms adapt to their environment? These aren't new questions. When Darwin first conceived of the concept of natural selection through descent with modification, he imagined a tree-like diagram shown here that demonstrates how organisms could be related to each other over time. So the tree of life, as we call it, starts at the 1 circled at the bottom of that image. And as organisms diverge through time, these branches of the tree diverge from each other and produce what are called leaves or tips that represent the organisms that we're interested in understanding. And this tree of life, or the concept of the tree of life, is still one of the fundamental concepts of evolutionary biology and forms the backbone of everything that I do as a researcher. Things have gotten a little fancier looking over time, but the fundamental concept is the same. We now call this branch of evolutionary biology phylogenetics, and its primary goals are to model relationships between living things. And using that model of relationships, we can then test hypotheses using statistical models. And this allows us to integrate information from statistics, computer science, and all of the different disciplines within biology. So it ends up being sort of a very broad and interdisciplinary pursuit. So let's walk through a quick example. Say we want to understand how a frog, lizard, human, and fish are all related to each other. First, we might measure some characteristics of those organisms. Most commonly these days, that means extracting DNA from these organisms, and then sequencing that DNA to produce what we call an alignment. It's essentially just strings of the DNA from each organism. And thus each organism is represented by a row, and columns are specific nucleotides in the DNA of that organism. So differences in those strings between organisms can tell us about evolutionary change through time. And we then use some type of algorithm and computer program to model how those evolutionary changes in the DNA have occurred, and that allows us to present some type of inference about the evolutionary relationships between these organisms, which we represent as a phylogeny. And phylogenies are these extremely information-rich diagrams. They show a series of nested and hierarchical relationships between species. So in this example, I'm showing a tree of our frog, lizard, human, and fish. And some important things to note about this diagram are that right now, the length of these branches there's no necessary meaning to the length of the branches. The only thing that really matters is tracing back where organisms have common ancestors. So we can see here that the lizard and the human share a common ancestor here, meaning that they are more closely related to each other than either of them are to the frog. And so that may be counterintuitive. You may look at this diagram and want to read it from left to right. And you might then assume that the frog and the lizard are more closely related to each other than the lizard is to the human, but that's actually incorrect. So I can show that here by just flipping the order of those two tips, the human and the lizard. This diagram shows the exact same set of evolutionary relationships. I've just rotated the order of that group. And we call groups and phylogenies clades. And clades tell us important information about groups of organisms that form these evolutionary groups. So a clade descends from a single common ancestor and then has diversified over time. So to think back to the bird example from earlier, all of those Hawaiian honeycreepers form a clade. They diverge from a single common ancestor. Now, these diagrams also allow us to add even more information. So, for example, we may want to display information about our belief in the probability of these relationships. We've used a statistical model to build these phylogenies. And so we can display some amount of uncertainty via posterior probabilities or bootstrap values at the nodes. So that's these are the nodes here, to show how much belief or certainty we have in these relationships. So here we're very certain that humans and lizards share a common ancestor. But as we go back in time, we become less certain about whether fish are related to this clade over here. Other types of information that we can include on phylogenies include characteristics about these organisms. So this is a toy example. I study plants, so I don't actually know much about frogs and fish, but I do know that they tend to like water. So here I'm just showing a sort of toy example of blue indicate that these are maybe aquatic organisms or water loving. And then the orange indicates that these are terrestrial organisms, or that they live on land primarily. And so I've gone through and said, for each of the tips in my phylogeny, each of these organisms, whether they are terrestrial or aquatic. And then we can use information about these evolutionary relationships to infer what the common ancestors of those organisms may have been. And this is generally done through other statistical models that use the information about these relationships. So we can see here that if both the human and the lizard are terrestrial and they share a most recent common ancestor with each other, it seems fairly intuitive that the common ancestor of those two organisms may also have been terrestrial. We can fill in the rest of the diagram this way as well, and infer that perhaps the common ancestor of all of these organisms was aquatic, and that there was a single event that happened right here that was a transition from aquatic to terrestrial. And this was just me making up a toy example and filling things in as I wanted, but normally this is done using statistical models that try to model the process of trait evolution through time and incorporate the information about the relationships between these organisms. Those models often use not just the relationships between organisms, but actually the amount of evolutionary time or the amount of evolutionary change. So the previous examples of these trees that I've been showing you, the branch links were meaningless, but often we do look at diagrams or phylogenies where the branches represent either some amount of difference between organisms, either a molecular difference, so changes in DNA, or morphological differences. And so that's shown here where these organisms, now you can see all of the branches do not come to the same point. And so maybe there's been slightly fewer changes on the branch leading to the human than there have on the branch leading to the lizard from the common ancestor of those two organisms. Often what we're most interested in, though, is the amount of time that's passed. So we want to know when was the most recent common ancestor of humans and lizards. Or going back to the albatross and coral example, that's how that inference was made of 800 million years of evolutionary change between those two organisms by building a phylogenetic tree of animals and using information to try to estimate how much time has passed since those two animals shared a common ancestor. So with all that being said, where's R? So I made this quick plot in R showing how researchers are using R in R packages to do things with phylogenies. So there's a list of all of the phylogenetics related R packages on CRAN, and then I added in some from other repositories such as Bioconductor, and then picked just the 10 most popular and core R packages, but the list includes over 80 R packages. So there's really this proliferation of different R packages to accomplish different tasks in R related to the whole pursuit of phylogenetics. And a couple of things to note here I think are interesting is that there's generally been an increase in the number. So these are number of downloads of R packages, and then from 2016 to the present. And so you can see that overall through time, there's been a general increase, perhaps with a slight leveling off towards the present, but it's hard to say. There's a lot of fluctuations there, especially in that top most dominant package, ape. And these packages do a bunch of different things. Some of the most popular ones are related to reading and parsing tree objects. So because phylogenies are these hierarchical nested relationships, the presenting that information in R requires a sort of, you can't do that in just a simple data frame. It requires a special set of, a special type of data object. So there's packages related to that, packages that allow you to reconstruct those evolutionary relationships. So to build that phylogeny or build the tree. And then there's a lot of packages that allow you to analyze data on those trees. So doing the type of what we call ancestral state reconstruction that we did with the aquatic and terrestrial traits. And then there's especially recently been an increase in packages that simply allow you to plot trees and associated data. And that's where my R package comes in. So I did a quick Twitter poll for this talk, just querying my Twitter community about how they use R in their work. So you can see that over 90% of the people who responded to this poll, so 90% of the people in my personal Twitter community who do phylogenetics use R. So R is extremely dominant in this field. Most people who do phylogenetics are using R in some way. And of those, the vast majority are doing what we call comparative analyses. So they're not building the phylogenies, they're not reconstructing evolutionary relationships in R. They are using those phylogenies to infer things about evolution, such as the terrestrial versus aquatic case. And that is probably because of computational limits for actually building these phylogenies. Most people end up using standalone software or other programs that are run in supercomputing environments, and R isn't necessarily optimized for that. But R is not the only tool that's being used. Some other languages that are popular. There is some support for doing these types of analyses in Python through a DendroPy package library. More recently, there's been some move to use Julia for some of these analyses. And there's also a separate language called RevBase. And RevBase is a probabilistic graphical model language, and it is a coding language that was created just for phylogenetics. So it's highly specialized, and it provides an environment for statistical computation based on probabilistic graphical models, and it allows for really specific coding infrastructure for things like Markov chain Monte Carlo and the tree-like structures. So aspects that are sort of fundamental to the task of phylogenetics that are not necessarily well supported in other languages. So let's go back to our diagram here of how we build a phylogeny. So we ended previously on some type of computer software, but what does that actually end up looking like? You'll see here that I actually put the RevBase logo on that computer. So if you run, you've extracted DNA from your organisms, you've sequenced your DNA, so now you know these strings of A, T, G, and Cs, and then you use RevBase to run some type of phylogenetic analysis, and this is what you get. So RevBase is extremely powerful, but it has certain limitations, especially for sort of your standard biologist who's not a trained computer scientist or a statistician, because the outputs are often these rather intimidating text files that contain samples from a Markov chain Monte Carlo analysis. And so one of the limitations to using RevBase has been that the output formats are different than other programs. They're not easily interpretable. There's no support for graphing or plotting your results in RevBase itself. And many of the sort of existing standalone graphical programs for phylogenetics are not capable of dealing with these types of outputs or are not optimized for them. So that's where our package came in. So some colleagues and I have developed Rev Gadgets, which provides a set of processing and plotting tools for visualizing the results of RevBase analyses. And we just put this on CRAN a couple of weeks ago, so it's still pretty new. And I'm just going to run through a couple of examples of some of the main types of use cases that we have, and then describe a bit about our next steps. So because one of our fundamental goals was to try to provide support for people who are not computer scientists or expert coders, we really wanted to make our code as easy to use and intuitive as possible. And so our main framework for the package was to create a set of paired functions that would allow you to, for a certain type of plot, read in your data file and process it, and then produce a publication ready, or really just kind of out of the box, nice looking presentable figure without having to do much else. So for example, here, we first read in the output file. So where it says files and then chromevol simple log, this is reading in the log file. So it would look just like that text file I showed earlier, just a bunch of numbers in a text file. And it reads in that file and processes it and calculates some summary statistics and saves that as this trace object. So that's what read trace does. Converts it to an R-friendly format. And then plot trace is a function we wrote that will plot the posterior distributions of these parameters. So what we're showing here are the posterior distributions of two parameters of our probabilistic model, gamma and delta. And in this example, not to get too in the weeds about it, but it's a model of how chromosomes change through time, chromosome number. So some organisms in animals, chromosome number tends to be fairly stable. But in plants, for example, chromosomes can kind of just do whatever they want. There's plants are notoriously liberal with changing their genomes. And so these two parameters are the gains of chromosomes. So you just an evolutionary change where you end up with one more chromosome than your parent. And then delta is the loss of chromosome, so where you lose a chromosome, one less chromosome than your parent. So that's what we're showing here is our estimates of the rates of gains and loss of chromosomes through time. And what's neat about this is that we're providing just this sort of R format for people to be able to read in their data and show these distributions. And they may or may not be interested in using this publication or using this figure in a publication, but understanding and looking at these posterior distributions is really important to understanding how your model performed and what type of inference has been made. And just staring at that raw text file is makes it very difficult to look at that type of thing. So we do try to provide the same structure with these paired reading and plotting functions for all of the different functionalities that we provide in Rev Gadgets. So this is another example where we read in a tree file. So we read in a phylogeny, and we want to plot that phylogeny and show along the branches some information about a rate. So in this case, rate might be a little misleading. What we're really showing is evolutionary inference of optimal body size in, I think these are cetaceans or whales. And so what we can see is that there are changes in optimal body size of whales through time. And so those correspond to the different colors of those branches. And again, we have this paired set of reading and plotting functions. So we have our read tree functions, which just reads in a tree based on some file path. And then the plot tree function, where we specify that we want to plot our tree object, that we do not want to format our tip labels in italics. And then the color branch by argument is how we tell the function to color the branches of that phylogeny by some parameter. And that parameter is stored in our tree file. And then we can change certain aesthetic parameters, such as the width of the branches. In another example, here we're showing change in these evolutionary rates through time. And again, we have these processing and plotting functions. Here, most of this code is just specifying the file paths, which are simply, they're a little long in this case, but we're giving all of these different log files. So this is 4 of those cumbersome text files that then this process div rates function is reading in and processing into an R-friendly object. And then we simply say plot div rates and give it that processed object, and it makes this plot straight out of the box. And there's all types of different customizations that we can do. These, all of our plots are based on either using ggplot2 or the ggtree R package, which is ggplot for phylogenies. And so you can, or the user can modify any of these plots after the fact, or just by adding the plus theme or that type of thing to customize the plots if they wish. But we wanted to sort of provide a standard aesthetic for the default. Yeah, so these show that over 60 million year period, we're estimating rates of speciation, extinction, and diversification. And we can see that extinction rate has remained relatively constant over that time, but speciation has increased slightly. So we see a large increase in that diversification that started around 40 million years ago. And I think this is from a primate phylogeny, but I'm not sure. So if assuming that this is from the primate phylogeny, this is just saying that around 40 million years ago, we started to see an increase in the number of species that were being that were emerging in primates. So a little bit of a more complex example, but one thing that's become increasingly clear in phylogenetics is the importance of model adequacy tests. So we have these evolution is super complex, obviously, and we're using these models that have certain parameters, and we're trying to apply these relatively simple models to extremely complex natural processes. And so for a long time, there's been, the field has talked about which model is best. Should we use a model that allows speciation to vary through time or not? But increasingly, we are recognizing that all of our models may be terrible. And if that's the case, then we probably should not be making huge conclusions about those evolutionary processes. So one of the main features that Rub Gadgets allows, among other programs, is to do posterior prediction, where you simulate under your model and then compare the values produced by your simulations to your observed empirical data, so the actual data that you started with. And Rev Gadget, the R package, also provides a set of plotting tools to examine the results of those simulations. And so each of these plots represents a certain characteristic of your data that you think your model should be able to capture. And then you have a curve that shows your simulated data, and then the dashed line, which represents your empirical or actually observed value. So this is a example of nucleotide evolution, so changes in DNA through time. And the model that we've used to simulate under is the simplest model of nucleotide evolution. And you can see that it is doing a pretty terrible job. In none of these cases do the simulated curves include the observed empirical value. So a user could look at this plot and say, maybe I should reconsider making some big conclusions based on my estimates of from that nucleotide model, because it seems that that model is not doing a good job of capturing the type of process that is occurring in my data. And again, we have these paired processing and plotting scripts. So we have our process, post, pred, stats, and our plotting function, plot, pred, stats. And that produces a list of plots, which we then can display as many as we want. So the phylogenies that I showed earlier were relatively simple in comparison to the amount of data that you can end up displaying on a phylogeny. So here's a more complex example. Here I have a phylogeny of ferns, and you can see that this phylogeny is time calibrated, which is to say that the values of, or the branch lengths correspond to actual time in millions of years. So we've included a geological timescale on the y-axis there so that you can see what geological era or epoch these events occurred in. And you'll notice that a lot of these tips or leaves, the ends of the phylogeny, don't come to the present. And that's because this tree includes a lot of fossils or extinct organisms. And this analysis is a more complex method of building phylogenies that allows you to include fossils as tips in your tree and infer how fossils are related to the living organisms, and use that combined information to make better estimates of how old this group is and when evolutionary changes happened. So in this case, we've got the geological timescale, and then we're also showing for each node of the tree. So for example, the places where branching events happen, it's called a node. We have a bar that corresponds to the 95% credible interval of our estimate for that age. And then our posterior estimate of our probability of that node occurring is the color of the bar. So you can see that there's a lot more uncertainty in evolutionary relationships within this big group of fossils because all of those nodes are orange. So we have, excuse me. We have less confidence in in the evolutionary relationships in that central fossil clade compared to, for example, this upper clade with a lot of more recent and still living organisms, where you can see that the posterior probabilities of those relationships are higher in blue. And you may also notice that the width of these bars increases as you go farther back in time generally. And that indicates that we have less and less certainty about when things happened the longer ago that they happened, which makes sense because most of our information here is coming from the DNA of living organisms, and it becomes much more difficult to make precise estimates farther back in time, even when including these fossils. And here I'm showing an example of showing ancestral state. So this is reminiscent of the aquatic and terrestrial example from the small little frog and human tree, but this is a phylogeny of all mammals, and we are showing placental type as the characteristic. And don't ask me questions about placenta because I can't even pronounce some of those words. But it makes for a really beautiful figure, so that's why we use it. But again, you can see that the code for making this figure is just reading in the data file, specifying what those character states are. So that's where we provide the placental type, and then providing that processed data object and specifying that we would like to plot it as a circular tree. And it produces that. And for my last example, it's another tree showing ancestral states. In this case, it's what we call biogeography, where we're reconstructing where organisms lived. So in this case, we're showing a plant example of Hawaiian silver swords, and then we are trying to understand where the silver swords, the ancestors of the silver swords lived, on which island. And so we have as our states, or we color our nodes by the island that, or combination of islands that we think these plants occurred on. And you can see that we show the states in this case as a pie chart at the node. And so that's another way to represent our uncertainty about these estimates. So we can say that maybe for this node here, about 50% probability that it occurred in this dark blue, which is the Kauai Island state. But then we're also showing that there's a decent amount of probability of it having occurred in these other colors, so these other areas. And this code becomes a little bit more complicated. So there's a little bit more that goes into making this figure in terms of having to adjust things for the aesthetics, but still compared to some of the other options, we've tried to give as much as possible, provide useful defaults so that the user doesn't have to fiddle around too much. So where are we going from here? We are thinking about, for the next versions of the package, trying to do more than just plotting the output. And so a colleague of mine has developed a super simple little Shiny app that allows you to put in Rev-based code, in particular for visualizing these prior distributions. And one of the most controversial and challenging aspects of Bayesian modeling is coming up with priors, especially for these evolutionary parameters where we may not have a very good idea of what our prior belief should be in those values. And so this little Shiny app allows you to actually visualize that distribution. And so especially for biologists who are not trained in statistics, it's not intuitive to think a gamma distribution and what that looks like and what that means about the values of those parameters. And we've also considered, this is a representation, a graph of a graphical model. And these can be really useful, especially for including in publications so that you're extremely clear about what model you are using. And so we've thought about a little widget that allows you to put in your Rev-based code into R, and it would produce some graphical representation of the model that you've coded. That is certainly future directions. So hopefully we can get that figured out. And with that, I'd like to thank my collaborators and Rev Gadgets, all of our dependencies, my institutions, and the North Organizing Committee. We'd be happy to take any questions.
Great. Well, thank you so much, Carrie. That was, for me, fascinating. I haven't studied biology since high school, I will admit, but it kind of inspired me. Visualizations were super cool, and I just admire that you're taking the time to think about those sorts of things to try to make your models more, easy to interpret, I guess, in a way. Yeah. So I have a couple of questions, but I'm going to see if anyone wants to raise their hand and ask a question, in which case I can unmute you. So if anyone has a question, do feel free to raise your hand or to type it in the chat. All right, maybe I'll inspire some questions. So so one of my questions actually had to do with the that sort of image you just had up on the screen, I think on the last slide. Yeah. So that image on the right-hand side, that was just sort of currently made by hand, if you will.
Yeah.
But you're working on a tool that would help make that image. Is that correct?
Yeah, that's correct. So this is one of the sort of examples of graphical models, but this was made I think probably in like draw.io or one of the other just sort of hand done graphing programs, but it does represent a model. And so one thing that, yeah, we would like to do is to be able to parse out the rev-based code that someone has run, and then say understand from the code what model they've specified, including what type of prior they've put on all of the different parameters, and then display that. One of the main challenges to that is that this little image here displays a pretty trivial model. And so they become really challenging to visualize in most cases where you have actually really complex. I've tried to do by hand for some of the models that I'm running. I've tried to draw out these diagrams, and it becomes like an entire PowerPoint slide of things with arrows crisscrossing, and so it's definitely it'll be a challenge to get that going, but I think it would be it could be really helpful.
Great. Yeah, that sounds, I mean, I'm just thinking about trying to write out model equations with, you know, sort of all those Bayesian parameters. And I'll admit I'm also not really a Bayesian, but yeah, that sounds complicated, and the picture just adds another level. Anyone else have some questions? Right, well, maybe I'll just leave it at that. I'm sure if folks have questions for you, Carrie, they will reach out. And I think we are on for another break. Our next speaker in this, our next speaker in this room is Adam Saffer, and he will be on at 12:30. So in the meantime, I'm going to play some more music, and you are welcome to take a break. I will also put a link to the other Zoom room in case any of you wanted to switch rooms at this point. I will drop that in the chat in just a moment.